Good afternoon, everyone. Thank you so much for coming. My name is Tanya. I will be moderating this series of seminars for the SymbNET Consortium. Just a very brief introduction about Nils. So he did his undergrad work in Bremen in Germany, and then his PhD work in the University of Saudi Arabia, King Abdullah University. And the focus of his research is the role of metabolomic interactions and the evolution of symbiosis. And without further ado, I think, Nils, you have the stage. Welcome very much, and thank you.
Thank you, Tanya, and thanks for the introduction and the opportunity to talk to you today. So I'm a postdoc in the lab of Anders Maipalm, who's also somewhere in the seminar room in case you have questions later. And for those of you who are familiar with Anders' work, you probably associate him with images like the one you can see here in the background. So nanosims is a technology that allows us to quantify and visualize the uptake of isotope tracers, and thereby we can effectively quantify and study metabolic interactions in symbiosis. And today I want to give you an example of how we use that to study, for example, the cnidarian-algal symbiosis and how it's regulated. And when we talk about this symbiosis, of course, we talk about it in the context of coral bleaching. So all of you are probably aware the symbiosis is having a problem. Corals all around the world are bleaching. They're expelling their symbionts in response to heat stress and other effects of climate change. And by doing so, they lose their main nutrient source. They starve, and as a consequence, these reefs are globally degrading and collapsing. And while we now for decades have very well characterized the environmental factors that cause this problem, what is surprising is that we still don't really understand the processes that cause this phenomenon. And so my own approach to solving this problem is taking a step back and looking at the larger scale of photosymbiosis. And when we do this, is we very quickly see that, well, it's clearly not a process related to host taxonomy. We see it in all kinds of symbiosis, regardless of host taxonomy, and the same applies to symbiont taxonomy. Even more interesting is that when we look at where these symbionts are actually located, for example, inside the cell, on top of the cell, completely outside of the animal, well, there seems to be no conserved pattern, and regardless of where these symbionts are located, bleaching still happens across all these symbioses. And last but not least, across all these symbioses, there seems to be no consensus of how symbionts are lost during bleaching. So we can degrade symbionts, we can expel them, and across all these symbioses, this tells us that when we try to understand bleaching, we can't approach this problem from a molecular level, because on the molecular level, well, across all these symbioses, bleaching has to be very different in each of these cases. And so if we just find one molecular solution, we still don't really understand bleaching as a whole. And so instead, we need to think about bleaching as an ecological property of these symbioses. And when we talk about the ecology of the symbiosis, of course, the basis of these symbioses is the nutrient cycling. And this is something that is very similar across all these symbioses. So we talk about it now in the example of corals, but the same applies to all of the other symbioses we have out there. And so the primary benefit in terms of nutrient cycling in the symbiosis is, of course, that we have a symbiont that is residing in or close to the host. And as a consequence, we have a heterotrophic host that provides a lot of CO2 to support algal photosynthesis. A part of this fixed carbon from algal photosynthesis, in turn, is translocated back to the host, where it fuels the host metabolism and in turn gets respired. As it gets respired, it turns again into CO2. So we have this nice efficient feedback loop where inorganic and organic carbon are constantly recycled in the symbiosis, thereby allowing to sustain very high productivity in the symbiosis. Of course, this only works as long as we have the algae willing to share the fixed carbon with their host. And currently, we believe the reason for this is the conditions these symbioses commonly occur in, which are oligotrophic. So we have severe nutrient limitation that limits the amount of carbon that can be incorporated into algal biomass. And the excess carbon, only the excess carbon, is then released into the environment, which in this case is the host. And so what we have is, in the end, severe nutrient limitation. In the case of corals, that is nitrogen limitation, that supports the algae to release carbon, thereby forming this efficient recycling loop that supports the symbiosis. And so the question is now, if this is the key to the ecological success of the symbiosis, what role does symbiotic nutrient cycling play in the maintenance and consequently in the breakdown of the symbiosis during climate change? And so that's the two components what I want to talk to you about today. First, the maintenance in the stable state, and then secondly, the breakdown during heat stress. We start chronically in the stable symbiosis, of course, but bear in mind that this part is what we're currently working on. So it's still a work in process. And if you think something doesn't make sense or is unclear, please tell me. It's still a very early stage of this project. And for this part, we use our beloved lab rat, Aptasia. It's a tiny sea anemone, which we can easily rear in the lab. And conveniently, it harbors the same algal symbionts we also find in corals. And so we can use this anemone to study the symbiotic interactions in the highly controlled conditions in the lab. And what we did in this case is we exposed this anemone to an environmental gradient of light availability, an exponential gradient from very low to high light. And of course, along such a gradients, we have trade-offs for the symbiosis. So in the low light availability, and of course, we talk about light limitation for photosynthesis. And then as we increase this light availability more and more, we talk about oxidative stress due to photoinhibition in the photosystem. And we can see that there are these trade-offs happening because at the very end of this light gradient, we see that on the one hand, we have symbiont mortality, these animals bleach, the algal symbionts slowly die. And on the upper end of this light gradient, we see that the host retains the algae, but we actually have host mortality. The host slowly starts to crumble away. And so for the first part, what we're interested in is now not what is driving the extreme limits of the symbiosis, but what is happening in between, what is happening in where we can maintain a stable symbiosis. How is this regulated? And we see that something interesting is happening here when we just look at the phenotype, because one could expect that we have some kind of linear response where we go from a host is benefiting the most to symbiont is benefiting the most kind of gradient. But what we see actually is a more symmetric response where we have, when we look at chlorophyll fluorescence in this case, highest pigmentation at the lower and at the upper end of the tolerance limit, and lowest pigmentation in the intermediate light levels. And so the question is now, what is happening here? And in terms of nutrient cycling, of course, the first thing we look at here is then the overall nutrient availability. And I apologize for the color scale. I just realized green and red is maybe not convenient to some of you. But what you're roughly seeing here, I hope all of you, is that we have some kind of optimum curve. So we have the CN ratio, the amount of relative carbon availability in the symbiosis, in the host and the symbiont in relation to nitrogen. And we see that we have this optimum of carbon availability, so the highest carbon availability in the symbiosis at intermediate light levels, and that decreases towards tolerance limits. And we said before that these algal symbionts try to only share excess nutrients to their host. And we indeed see that when we look at the partitioning of acylated photosynthetic carbon via 13C isotope traces, in this case bicarbonate. And we can quantify using this technique how much of the assimilated carbon is assimilated by the algae and how much is translocated to the host instead. And we see that where there is most carbon available, these algae are also more generous in sharing their resources with the host, and towards the suboptimal conditions in terms of carbon availability, these algae are more selfish and retain more carbon for themselves. And so the question is, this selfish behavior, what does it mean for the regulation of the symbiosis? And what we see is that, first of all, this partitioning of photosynthetic carbon in the symbiosis directly regulates with the partitioning of the uptake of environmental ammonium in the symbiosis. So the more carbon is translocated by the algae to the host, the more ammonium from seawater eventually ends up in the host tissue. And the question is, why where is this correlation now coming from? And for this, we can no longer talk about bulk isotope assimilation patterns. We need some spatial context. And this is where nanosims comes in. So we get heat maps like this, spatial illustrations of isotope assimilation in a biological tissue here. And so you can see here the two cell layers of the host. We have the ectoderm, we have the gastroderm, and in the gastroderm, you see these algal symbionts, and they're deeply pink, telling us they assimilated a lot of 15N ammonium under optimal conditions here. But we also see that there's some enrichment of ammonium uptake in the surrounding host tissue. And so now one way to look at these data, of course, could be that this uptake of ammonium here is the result of translocation. So these algal symbionts are autotrophic, so they assimilate ammonium, and they not only share carbon, they also share nitrogen with their host. But we see that this is actually not what is happening when we repeat the same study in the presence of pyruvate. So a carbon backbone, and we can see that the carbon is utilized by the host to now, throughout the entire tissue, assimilate ammonium into its own biomass. And by doing so, we see that we have more ammonium uptake in the host tissue and proportionally less uptake by these algal symbionts. So the host and the algal symbiont compete for the same nitrogen source, and the more the host assimilates, the less is available to these algal symbionts. And so we are here talking about a direct competition in terms of nutrients, in terms of nitrogen between host and symbiont. And what is interesting about this concept is that we often think about symbiosis in a kind of reward and penalty system. So good behavior gets rewarded by the host. But here we actually have the opposite. The more the algal symbionts are sharing their resources with the host, the more the host punishes them by retaining environmental nitrogen from them in the symbiosis. And we can see this punishment in a way when we look at the population density. So the more carbon is being shared, the lower the population density of these algal symbionts in these animals. So we really have a competition between host and algae here for available nitrogen, and the outcome of this competition is a consequence of how much carbon is being shared in the symbiosis. And now this competition, of course, explains what we saw in the beginning. We have a U-shaped curve in terms of symbiont density, where at suboptimal conditions, the host is not able to keep these algal symbionts nitrogen limited, and we have higher population densities towards the tolerance limits of the symbiosis. These symbiont densities directly translate into chlorophyll content, and that in turn explains this phenotype we saw in the beginning, where we have highest fluorescence towards the edges. And so what we see here is basically that the symbiosis is maintained by the competition between host and symbiont, and as a consequence, the relative fitness of host and symbiont is in kind of an inverse relationship. While it means that we, of course, can have mutualistic interactions in some way as a consequence of this competition, there's a very narrow ecological niche where neither partner outcompetes the other one in this interaction. And of course, when we have a narrow ecological niche, this is always a problem in the context of climate change and global environmental change. And so this is where we get to the second part, now the destabilization of the symbiosis in terms of heat stress. And for this, we actually switched to corals now. So we used the coral Stalophora pistillata, and we did that during my PhD in Saudi Arabia at Coast University, where thanks to my colleague, we know the local reefs really well. And for example, we do know the environmental conditions of these reefs, where we can then use this information to extract properties for regular non-bleaching years, such as the mean temperature and the annual absolute maximum these corals ever experience. And we use this information to design our aquarium study, where we expose corals for 3 weeks to either the mean annual temperatures or the absolute temperatures of such a year. And we see that these corals in the Central Red Sea, they're already living extremely close to the thermal limits, because what we see is that while we take temperatures from a regular non-bleaching year, if we maintain these peak temperatures long enough, we eventually do get bleaching anyway. So it's a matter of how long these peak temperatures are maintained here. But of course, in terms of understanding the breakdown of the symbiosis, this time point is kind of uninformative because, well, the breakdown has already happened, the symbiosis has collapsed. So instead, what we do is we channel our energy and resources into the earlier time point where we, from a visual phenotype, still have a stable symbiosis. We have no changes in symbiont density, and we have no changes in chlorophyll content. And so we focus on this time point now to really characterize what is happening inside the interaction of the symbiosis prior to the actual breakdown of this, prior to its breakdown during bleaching. And to get an idea of what is happening first, of course, we look at the transcriptomic response of host and symbiont, and clearly what we didn't find was the expected responses such as oxidative stress, all the well-described markers of bleaching, because at this early stage, this is not what is driving the response. Instead, what we see is predominant effects of energy metabolism and nitrogen metabolism. So predominantly, we see a downregulation of anything related to energy availability, such as, for example, the TCA cycle in the host. Closely linked to the TCA cycle and energy availability as a whole is, of course, the amino acid metabolism. So we have the glutamate metabolism, which uses substrate from the TCA cycle, as well as energy to fix ammonium into glutamate, or the other way around, it can use glutamate as a substrate for the TCA cycle. And what we see now is that during heat stress, we have, as a consequence of energy limitation, less fixation of ammonium into amino acids and an increased degradation of amino acids to fuel the TCA cycle as an energy substrate. As a consequence of that, of course, we have reduced ammonium uptake and an increased release of ammonium in this metabolism. On the alga side, on the other hand, what we see is no evidence of this energy limitation. We have no changes in TCA cycle or amino acid synthesis for that matter. However, what is interesting is that we have this stable amino acid synthesis despite clear reductions in anything related to nitrogen uptake, such as the uptake from the environment or the generation of nitrogen via catabolic activity. So the question is now, where is this nitrogen coming from if there's less investment into the uptake of this for the cell? And for that, we now need to look at the symbiotic nutrient cycling. And we start by first carbon cycling on the holobiont level, so photosynthesis, how much carbon is available. And to our surprise, we see that the energy limitation in the host is not a consequence of algal photosynthesis. Instead, what we see is that we actually have more photosynthesis during heat stress, which is, of course, explained by respiration. If we have simple metabolic activity as a consequence of temperature, we have more respiration during heat stress. More respiration means more CO2. So we fuel photosynthesis during the light. However, any excess released CO2 in the light is not recycled during the dark. So at night, whatever is respired is effectively lost from the symbiosis. And as a consequence, we have this increased net deficit in carbon in the symbiosis during heat stress. And so the interesting part is now looking into what does this energy deficit mean for the respective symbiotic partners. And for that, we go back to nanosims. Here, in this case, we have the 13C assimilation, and we see, of course, that the most is assimilated in the algae itself, and we have a lot of translocation to the surrounding host tissue under control conditions. Now, during heat stress, however, the picture changes drastically. We still have a lot of assimilation by these algae, but we see much less translocation to the surrounding host tissue. And we can quantify that, and we actually see that we have less translocation to the host and more retention of photosynthetic carbon by these algae. And when we talk about more selfish behavior, we already now know that this is linked to nitrogen cycling in the symbiosis. And indeed, what we see is on the holobiont level, we have the shift from net ammonium uptake to net ammonium release. drastic net ammonium release actually, which is of course directly linked to what we saw in the transcriptome. So the shift from amino acid synthesis by the host to amino acid degradation, thereby releasing excess ammonium. And this is what we see on the holobiont level. And this directly affects, of course, the nitrogen availability for each symbiotic partner. And we can see that again in the nanosims. So same as before, under stable conditions, we see assimilation by the algae and in the surrounding host tissue. But during heat stress, this assimilation completely or largely disappears. Of course, as a consequence of this intrinsic ammonium generation. So if you have a lot of ammonium intrinsically being generated, you have no longer any demand for environmental nitrogen from the outside, and we see that in both symbiotic partners. So clearly this reduced uptake of ammonium from the environment does not mean that there is effectively less ammonium available. Instead, it's quite the opposite. You can see that when you just look at the shape of these cells, here we have a nice regular cell, but here we have some kind of weird distortion. And what that is, of course, is simply the difference between a regular and a dividing cell. And so we can use these nanosims images now to not only quantify where these nutrients are distributed, we can also get an idea of how many of these cells at any given state are dividing. And so we can come up with a kind of mitotic index. And we see that during heat stress, we actually have 3 times the amount of dividing cells in the symbiosis. So indeed, more nitrogen is available to these algae. They are no longer nitrogen limited. And as a consequence, they can invest photosynthetic carbon into their own growth rather than translocating it to the host. So coming back to our overall nutrient cycling, what does that mean? During heat stress, probably the first response we see is increased metabolic activity of the host and increased carbon demand. And this, in part, is fulfilled by using any biomass energy reserves the host has, and also by cutting down on synthesis of new biomass in the form of amino acid. As a consequence, we shift from a net ammonium uptake to a net ammonium release in the metabolism, which in turn increases the nitrogen availability for these algal symbionts. Happy to be deprived of the nitrogen limitation, these algal symbionts can now invest their photosynthetic carbon into their own growth rather than translocating it to the host. And this, of course, ends up in a nice positive feedback loop where we can reiterate these processes over and over again until we reach this point where these algae have so much nitrogen available that there's no longer any incentive to translocate to the host. And of course, if there is no translocation to the host, that means that during heat stress, eventually we reach this point where there's no longer any ecological benefit of hosting algal symbionts for the host. And if this is the case, then of course, we still can't explain bleaching from a molecular level, but we can now explain bleaching from an ecological level. And so we can explain really the breakdown of the symbiosis as a consequence of this feedback loop. However, for the symbiosis to ever be stable, we can't have just one feedback loop, so we need to also stabilize it by an opposing feedback loop. And this is what we kind of see in our data as well, because we see we have this pronounced proliferation of algal symbionts. And we can now assume that the more these algae proliferate, well, eventually there's more and more algae competing for the same amount of nitrogen again. And so there is a potential for this feedback loop to counterbalance this feedback loop. And it's the competition of these two opposing feedback loops that determine the trajectory of the symbiosis in the end. So during bleaching, clearly, what we see here on the blue side is dominating. We see this shift of the symbiosis from a new nitrogen limited state towards a carbon limited state where there's no longer any efficient recycling of carbon in the symbiosis. However, in reversed interaction, the same process can also explain us how we can switch from a carbon limited state, such as, for example, the symbiont-free animal, and explain the establishment of the symbiosis by the algae through rapid colonization as a consequence of high nitrogen availability. eventually leading to nitrogen limitation, pushing the symbiosis towards a stable state where we have a mutualistic nutrient exchange in the symbiosis. So all of this together, what I think, there are 3 points I think we need to take home is, first of all, clearly the symbiosis is a mutualism, but it arises from the competition for resources between host and symbiont. The relative competitive advantage in the symbiosis is a consequence of environmental conditions. And thereby we can use symbiotic nutrient cycling to explain and predict the onset, the maintenance, and the breakdown of the symbiosis in light of the nutritional status. And with that, I just like to thank all of you for listening. Big thanks to all the co-authors and supervisors, and also all the people that helped with the studies. And I'm happy to answer any questions.
Thank you so much, Neil. It was a very interesting talk. Questions for Neil? Please unmute yourself and feel free to ask. We have time for a couple of them. Or you just can write them in the chat. So if no one steps up, Niels, I was just wondering, when you do this on the last system that you showed, did you ever try, I mean, is it even possible to keep this system in an artificial environment? So can you stimulate different, not only different temperatures, but like different salinities of the environment, for instance, so that you can simulate different conditions?
So in terms of, so the second part was on corals, really, and sure, I mean, there's thankfully corals are quite popular in aquaristic trade, so we have immense data availability from all these hobbyists trying all sorts of things by accident on purpose. But sure, you can modify salinity in aquaria, you can modify light and everything, but the problem with corals is they're incredibly sensitive and very unstable, as we unfortunately all know. And so there's a very small tolerance range where you can play with. And so this is why the Aptasia, our sea anemone model, is also a bit more informative, because we still see the same processes, but in a much wider tolerance range, because simply there's a bit more metabolic flexibility. And so we have a more leverage to expose more extreme gradients that we can then get a more easy resolution in terms of these responses.
Yeah, sure. And did you ever look into the algal genetic diversity? So I don't know how conserved or how much variation you would expect in this algae, because I assume they divide by mitosis, right? So have you ever studied if there's any kind of genetic mutations that would make them better or worse symbionts?
So in corals, we know that within one host colony or individual, normally there's only one genotype dominating, because, well, there's colonization by the same genotype simply through asexual division. However, what we see in terms of geography is that, like, in different ecoregions, we see clear patterns of different symbionts dominating different regions. And now there's a big question of where this is coming from. On the one hand, of course, we have different genotypes that have different susceptibilities to temperature, but there's also, and I think this is more clear in the data, is that we have different concentrations of nutrients in different environments. And so, for example, we work in the Red Sea, which is one of the most oligotrophic systems, and we find completely different symbionts to, for example, what we see in the Great Barrier Reef, where we have much higher nitrogen limitations. And these, simply because now we believe that bleaching is a property of nutrient cycling, these intrinsic differences in adaptation to local nitrogen or nutrient conditions also translates into some differences in terms of bleaching susceptibility. And for example, we can see that corals in the Red Sea on average are much tougher than anywhere else where we have higher nitrogen nutrient availability.
Okay, this was great. Thank you so much, Niels.